that sound okay? Well, anyway, yeah, it's really great to be here talking about this eDNA survey that we did a few years ago. Anyway. And I'm going to start by introducing you guys to Knight Inlet, which is a glacial fjord sort of northeast of Vancouver Island in British Columbia. It's a really interesting place. It's long, so it's about 100 kilometers long, yet only less than 3 kilometers wide in its widest place, and 500 meters deep in its deepest area. And so it's this really cool, unique biodiversity hotspot where you have super deep water organisms that live really close to coastal ecosystems. And in particular for today's talk, I'm going to focus on the Hoya Sill, which is an underwater ridge right about a third of the way up. And it's really unique because it comes up to a shallow depth of about 65 meters is the deepest, but it comes even shallower to diver depth. And so at this Hoya Sill, you get these really deep water species existing at diver depth. And so it's this totally unique ecosystem in this nearshore habitat. And so just highlight some of these really cool sponges, basket stars, these deep water corals that would usually only be hundreds of meters deep occur 50 meters or even at diver depth there. And then also it's notable for these large healthy populations of sunflower stars, which have been decimated due to sea star wasting disease. So it's a really cool place. And so we were approached by the Mamalakula First Nation to carry out a biodiversity assessment of this region in support of their work towards making it an indigenous, protected and conserved area. And so we worked with them to design a biodiversity survey that could then support their management and conservation goals. And so really briefly, they just selected the areas within Hoya Sill that they were curious to have a good diversity survey. And then within those areas, so we picked 6 different sites where we paired eDNA and a roving diver survey. And we collected eDNA surveys from the surface, the midwater, and the bottom, and then we're able to compare that to visual surveys of benthic diversity. And I won't get into eDNA too much, just to say that in this talk, so I'm talking about eDNA collected from filtered seawater samples, and all of the eDNA that I talk about will be metabarcoding or amplicon sequencing. And a little bit of the methods, because I think people might be interested. At each of those depths and at each site, we use triplicate seawater samples plus field blanks on board the boat. And we use these gravity bags for these field surveys in remote locations. And so these are basically IV bags that the divers can bring down to depth, open and fill with seawater, and then bring them up to the surface, and then we just hang them up and have gravity filter them through the Starabax filters. And so we found it's a really nice approach, and it's a pretty cheap approach for doing surveys in remote areas. And so we use 2 markers for the metabarcoding, the Lemay CO1 primers and the 12S for fish. And then I'll just say, so both the dive surveys and then our molecular approach, we binned everything into 3 taxonomic groups. So we're getting the fish from the 12S, using CO1 for the marine invertebrates. And then also, I'm sure people who have used the Lemay primers know that they're very conserved and you get a lot more than marine invertebrates. And so we also retained all the algal ASVs in a separate data set from the Lemay data as well. My wife is a seaweed taxonomist, and so I always keep algae data around. Yeah, so I'm just getting into the results. Starting just with an overview of the fish data from the 12S. So the pattern that we see with these data, I guess, is that all of the surface samples contained quite unique biodiversity compared to the bottom and the midwater samples. And then within depth, we found clustering by the site. But certainly a really strong pattern of different diversity recovered in those surface samples. And that was carried through to each of the 3 different taxonomic data sets that we looked at. So for the fish, inverts, and algae, the samples that we recovered at the surface were quite significantly different in terms of their communities from what was deeper down. And then, so just to dig in a little bit into what's driving those patterns, for the fish, it was a pretty overwhelming pattern that our surface samples were dominated by salmonids. We found most of the Pacific salmon species present in these samples, whereas deeper down, we saw lots of herring and also some greater signal of rockfish, plus a whole bunch of other species. But certainly the abundance of salmon in the surface samples really drove those patterns. And then similarly, just to show the invert data, again, strikingly different communities in the surface. Rotifers dominated the communities in surface, whereas copepods and other zooplankton were the most abundant taxa that we recovered in the mid and the bottom samples. And so there haven't been enough Venn diagrams, so I thought I'd throw one up here. Just to compare what we're seeing in the eDNA versus the visual surveys that the divers carried out. And I think that's an interesting comparison because it's not random. Like there's actually some ecologically interesting patterns that result from this comparison. So those salmon that we saw dominating the surface samples were absent from the diver surveys, as were the herring, which are really abundant in all of our eDNA data. Conversely, the visual surveys picked up a lot of demersal species that were rare and heavily affiliated with the bottom that weren't making their way into our eDNA samples. And then, I should just say, sorry, also the really important thing is that we also recovered many more species in our eDNA than we did in our visual survey. Is that working? How's that? Yeah, we certainly, in terms of species level identifications, our eDNA had much greater diversity than the visual survey. And so I like to compare that to the data from the invertebrate survey. This one's interesting because I'm only including species level identifications. And so from this perspective, the visual survey, in terms of richness, outperformed eDNA because we weren't able to get species level identifications for a lot of the ASVs in the data. Certainly there were thousands of ASVs that were present, but in terms of species level annotations, it wasn't as good. And also it's quite diagnostic. We're seeing things that a diver is never going to see, so a lot of zooplankton and a lot of small cryptic species, whereas the divers were noticing more typical benthic invertebrates. And so I wanted to comment a bit on annotation success rate, since it's clearly a factor in that last Venn diagram. And so for this figure, there's 3 panels for the fish, invert, and algal data sets respectively. And so I've just thrown a bar up for each taxonomic rank. Red means the ASVs were assigned, blue means they weren't. And so the point I wanted to make with this slide is just that for fish, basically everything can get annotated to species, so there are maybe 90% annotation success rate to species for the fish. But when we get to the CO1-based data, we're at less than 50% of ASVs that we can annotate to species level currently. And so we're doing a lot of DNA barcoding and a lot of work to try to build up the reference databases, especially for our local species, but certainly that is a shortcoming. So just to conclude, I want to say that I think from our survey, and especially for these coastal fjords, if you're budget limited, I think we can maximize diversity by adding more depths and more stratification of samples rather than just more sites of the same depth. Clearly reference databases need a lot of work. And also I think that this is a nice example, again, of eDNA and visual surveys being complementary. They're both detecting diagnostically distinct diversity. And I also want to end by just saying that after we carried out this survey, the Mamalakula declared this region indigenous, protected, and conserved area. And now they're working with the federal government to make it recognized by the government of Canada as well. And also in 2022, we did a follow-up survey. So again, they identified more regions or more sites within this region where they wanted additional biodiversity data. And so we're adding, in addition to eDNA, some more ROV surveys, multi-beam surveys, and also a more quantitative diver survey as well. And so we're continuing to work with them to help with this indigenous protected area. And with that, I will say thanks. And